Okay, so yeah, I just mentioned if you want to make a meeting with me to talk about your study ideas for group project 2, please don't hesitate to do that. Today we're going to talk about, I have analyzed the data for group project 1. We ended up running apparently 76 participants. I will show you a little bit of how to perform that data analysis. And I will kind of go through the interpretation of the results. I have finished the instructions for group project 1, second half. So it's already posted on Blackboard in the group project 1 folder. I've also created a link to the instructions as well. We'll go over those. I've made the due date for that November 1st, which is a Sunday at 11:59. And I'll point out that the second half, you're writing the method section, the results section, and the discussion section. Oh, in addition, I haven't mentioned this before, but you'll be finishing the abstract. You'll write an abstract, which is a summary of the entire paper. So we'll go through that group project 1 stuff. Then we will talk about the EEG stuff that we started last time. And I believe we'll have time where we'll be able to talk a little bit about the article. You have a quiz for this article, which is due tonight. And I'm going to be giving, assuming we do get to talking about it today, I will be giving you some information which is relevant to the quiz, which is due tonight. I will also point out that we have 2 more EEG related quizzes, which I will post today. EEG1 and EEG2. I have made both of those due, I believe, Saturday at 11:59. So that is the plan for today, is talk about these things right here. So, and I should also mention that for today's slides, all I've done is I modified the slides that I used last time. I've added a couple things in the middle and actually towards the end. So my suggestion is to go back to Blackboard, and the slides that we used last time, I've re-download those because I've updated them, including some new things, which you'll probably want to take a look at. Okay, so let's get into this here. Yeah, I showed you this one last time. I'll just point out, when you're writing your method section, you're going to need this information about the participants. And actually, let's go to it real quick. This is the participant info. And if you add up all of these things, it's 38 plus 10 is 48, 48 plus 28 is 76 participants, which is correct. There was an error though, so I'm a little bit confused, but it's not the end of the world. This one participant, apparently participant number 142, was counted as being both a SONUS system participant and a friend and family. So I'm a little confused about that. That doesn't seem like it's possible. Because if you run a person in SONUS system, then you don't count it as a friend and family. The friend and family is only when you, someone that you ran that did not use SONUS system. So that's an error. If the group that wants to fix that, they can. But at this point, I don't necessarily want to spend a lot of time troubleshooting that one particular error. It appears that there were 76 participants if you add all this up. And this goes with, if you click here, this is the actual data. We'll scroll down here all the way to the bottom where we summarize the data from group project 1. And if you add, there were 40 participants in the Mozart condition, 36 in the rain chimes condition, which equals 76. So although there was that error in the participant spreadsheet, looked to be an error, the total there was 76. The total here was 76. So it appears that we ran 76 participants, 40 in the Mozart condition, 36 in the rain chimes or the control condition. And this is the type of information that you will want to include in your method section. And you'll want to mention in the method section that 38 of the participant, or you don't necessarily have to say the specific numbers, but you can say over half of the participants will run through SONUS system. The small amount of participants will run extra credit from a cognitive neuroscience class at KU, and around 30 participants or so were run from who are just friends and family of the participants. So you want to provide the information. Basically, I don't really care if you give the specific number of each of these, but you'll want to make it known in the method section. Some of the participants were from SONA system. Some of the participants were receiving extra credit from a cognitive neuroscience class at KU, and some of the participants were the experimenters, friends, and family. I'm hesitant to tell you to say the specific numbers because I'm not actually for sure that these specific numbers are right, in particular because of that one error where one participant was counted as two different things. So these numbers are probably not perfectly accurate. So you don't have to say the specific numbers, but do mention that there was 3 different kind of ways that we sampled participants. You can also mention that different groups, there were 7 different groups and each group ran about 8 participants. So you can mention that there was basically 7 different groups of experimenters that were each collecting parts of the data. They're collecting data from part of the subjects in the study, part of the participants. Okay, so it looks like that stuff's done. I will show you the data analysis for that here briefly, or here shortly. And stuff I showed you last time. Okay, so here's where I'm going to show you how to analyze the data of using SPSS for our study. So first off, I've created a new Google Sheet right here. And I've created a new Google Sheet where instead of all the data being like really spread apart, like one participant here and another participant down here, another participant here, I've pushed all the data up to the Google Sheet so everything is nice and organized. And I can show you that now. Here they are. These yellow ones so here are the data. The Mozart condition is over here. The rain chimes condition is over here, incongruent, congruent, incongruent, congruent. So it's the same basic structure as the other spreadsheet, but I just put everything all nice and organized so that it would be easy to copy and paste into SPSS. I have highlighted some of these yellows, not necessarily because they were errors, but because the reaction times for these yellow ones were particularly long. For instance, this participant right here, it was taking this participant over 2 seconds on average for each trial. So this particular participant was spending a whole long time deciding what the correct answer was for each trial of the Stroop task, which we haven't really talked about excluding outliers, but in general, in reaction time studies, if there are participants whose reaction times are way slower than everybody else's, that there are potential situations when you might exclude the data. We're not going to do that for this project, but that's why I've highlighted these, because these are people that took a whole long time to respond. And there happen to be more of these people in the Mozart condition than the rain chimes condition. I don't know if that's just a statistical fluke or if there's something about the Mozart condition which makes people generally slower, which I don't think is the case because we did not find statistical evidence that supports that. But if you notice, for instance, this person is taking 2.3 seconds or 2,300 milliseconds, but then there are other people that were only taking like 500 milliseconds. So this participant was taking 4 times as long on each trial to answer the Stroop task. So it's kind of curious as to what was taking that participant so long. But nevertheless, I kind of yellow those for my own reasons. You do not necessarily, that doesn't affect the way you're going to do things. So what you're going to do is you're going to copy and paste 2 different times into SPSS. I've already done it. So you would, for instance, highlight all of the reaction times in the Mozart condition right here and press Command C. And then you would go to SPSS, and then you would copy and paste them right here. So incongruent, congruent, it's the same order. Incongruence on the left, congruence on the right. 1180.44, this should be the one who's at the very top of this. Let's go check to make sure it is. 1180.44 is this, the first number here, and you'll also notice that it's the first, oops, that's not what I want to show you. It's the first number right here. So the first 40 people, the first 40 lines, the first 40 rows in SPSS, I'll kind of highlight them here. This is the old one. Okay, so this is still good enough. The 40 people, these are the 40 people right here. These people were all in the condition Mozart. And I coded that, I dummy coded that with a 1 right here. So you have to create a new variable using variable view right here. Call it something like type of sound, or you can just call it sound if you want. And you'll do all 1s for anyone who is in the Mozart condition. And then right here, this is where you will copy and paste the rain chimes condition. So then you'll do it right here. Command C. Good. And then you have to dummy code this. So these are going to be all 2s. Should stop at 76. All right, good. So I want to point out just a general rule about SPSS. When I do this thing, this dummy coding where I put 1, 1, 1, 1, 1, 1, 1, that means these 1s mean this participant is in the Mozart condition. All the 2s means the participant is in the rain chimes condition. You only have to do dummy coding when you have a variable in a factor in your study, which is between subjects. In our study, in our group project 1, type of sound was between subjects. About half the participants heard Mozart. The other half of the participants heard something different. They heard rain chimes. So type of sound was a between subject factor. That's why we have to do that dummy coding. Then to actually run the analysis, let me check the output right here. Yeah, it looks like the output is the correct output. So to run the analysis, you would click Analyze, General Linear Model, Repeated Measures. And then I've already added the factor. So the within subjects factor in our study is congruency. No matter which condition, whether Mozart or rain chimes, everyone did the Stroop task. And the Stroop task has 2 conditions, congruent and incongruent. So we have 1 within subjects factor. It's already been added. I already did it earlier, congruency, and it has 2 levels. That's why the number 2. So I typed the number, I wrote the word congruency here, and then the number 2 here, and I clicked add, and then it showed up there. That's the only within subject factor, so we can click define. Now we have, we'll move incongruent right there, and we'll move congruent right there. And then here in between subjects factor, our between subjects factor is type of sound. So please note, this is how you set up a mixed design ANOVA. I think I've actually shown you how to do this before with one of the previous labs. I think the IAT lab that we did earlier in the semester was a mixed design as well. But please note that the levels of the within subjects factor are entered here, and then the name of the between subjects factor is entered here. Let's see if we need any options. Yeah, we'll include descriptive statistics. If you go to graduate school, you're always going to need to include estimates of effect size. I'm actually not requiring you to do that for the group projects in this class, or at least for group project 1. I might change my mind about group project 2, but in order to get eta squared, which is a measure of effect size, you would click that box right there of estimates of effect size. I'm going to leave that not checked for now. Just descriptive statistics is all we need. Then we would click OK. And let's make sure things are right here. Yep, things look good. So one thing that I always like to do when I run SPSS analyses is include those descriptive statistics and then look to make sure that the means that show up in the descriptive statistics box match what you calculated in the Google sheet. We don't have to, they look like they're right to me, but let's just check one of them, 873.3024. 873.3024. So there's a match. You want to do that for all of them. Make sure that the means that we calculated in the Google Sheet or Microsoft Excel match the means that show up in SPSS. And that means that you probably did the data analysis correctly. So I'm not going to actually show you the output right now. I want you to try and do it yourself. If you have questions about, I've shown you how to run the test and then you'll get the output. I want you to try your hand at interpreting the output by yourselves and putting it in correct APA format by yourselves. We've done lots of practice with APA formatting of F, but if you're not comfortable doing it by yourself, there's no problem with asking me, showing me your output, and I can help guide you through the different components, explain what things mean, and tell you exactly which part you have to include in your APA format when you're presenting the output. I will point out though that because this is a two-way, let's go back to the slides here. This is a two-way mixed design ANOVA that I just ran. We're going to have 3 F values that you're going to have to report in the results section of group project 1. So you'll have to include the F value, the degrees of freedom, the P value for the main effect of congruency. That is, are the reaction times between the incongruent and congruent conditions different? You will also have to include the F value, degrees of freedom, and the P value for the main effect of type of sound. So what this is asking is there an overall difference in reaction time between the Mozart and rain chimes condition? You ignore the factor of congruency and you just look overall, are people quicker or slower after listening to Mozart compared to rain chimes? And actually, I do want to show you a quick thing here regarding that. I was a little bit surprised. If you actually look in general, the average amount of time it took people in the Mozart condition was around 840 milliseconds. And the average difference in the average reaction time in the rain chimes, if you kind of take the average of these two, is around 740 or so. And so there's 100 milliseconds difference overall between these two conditions, though that difference was not significant. So it is not the case that people are significantly slower in the Mozart condition compared to the rain chimes condition, although there is like 100 millisecond difference or so in the overall reaction times here compared to the overall reaction times here. That was definitely unexpected, but it wasn't significant, though it was technically pretty close to significant. So that's the main effect of type of sound. And please recall, type of sound is the between subjects factor and congruency is the within subject factor. The interaction between those two factors, this was the thing that was really important for our hypothesis. We hypothesized, you didn't have to explicitly say this in the introduction, but basically what we hypothesized was a significant congruency by type of sound interaction. To explain, to kind of say it again, I wrote it earlier in the slides. Basically what we expected is that for people that listened to the Mozart stimuli, we expected the difference in reaction time between the incongruent and congruent conditions to be small. We expected that people who listened to the rain chimes condition, that the differences in the reaction times between the incongruent and congruent conditions would be bigger than the difference for the people that listened to Mozart. But we didn't find that. So yes, I want to do this. I'm doing, that's just the Price is Right sound when you lose the game. It's really not that big of a deal. Our hypothesis was not supported. And you know, it's a little bit sad, but it's really not the end of the world by any means. I think I've mentioned before that all of the empirical papers that I've ever published, my original hypothesis was never the exact way that the data turned out, but it still gets published. So just because your hypothesis doesn't work doesn't mean that the findings are not valuable and publishable. But ours was not. And you'll see that when you run the analysis, and you'll see that the p-value is not for the interaction, is not less than 0.05. So we don't have the significant interaction. And I'll also show you the data right here. So I've made a graph. I'm going to teach you how to make this graph Monday's class. And I'm part of the second half requires that you basically include this graph. So this graph is basically APA perfect as far as I can tell. I've included my little watermark so that you can't just steal this one and include it in your paper. So I really want you to try and make this yourself. I'll show you how to do it Monday. But you can kind of see basically what I've just described. You can see it pretty clearly when you're looking at this graph. So of course, correct reaction time is the dependent variable. These are the bars for people that listen to Mozart. These are the bars for the people that listen to the rain chimes. The black bars are the incongruent condition. The light gray bars are the congruent condition. And you can see that for both the Mozart condition and the rain chimes condition, no matter what you listen to, the participants were slower. That is, the reaction times were bigger for the incongruent condition compared to the congruent condition. So, you know, there's Stroop interference right there, the Stroop effect right there. Incongruent reaction times are bigger than this. Same thing for rain chimes. The incongruent condition, the reaction times are slower than for the congruent condition. So there's a significant difference right here. So we see Stroop interference right there. we see Stroop interference right here. But when we're trying to interpret that, when we're looking at that interaction component in SPSS, it's not significant. And we're trying to understand what would it look like if it were significant? What would the data look like if it was significant? And since the data are not significant, like how could we see the fact that the interaction is not significant in this graph? And so if our hypothesis was as we were expecting, then the difference in reaction times here would be very big. And the difference in reaction times here would be relatively smaller. That's not what the difference is. There might be some slight differences here that are marginal. There's obviously a difference here, and there's a difference here, and the difference between the Mozart and the range times bars might not be the identical same difference, but the difference is not significant. The interaction is not significant. The difference here is basically the same as the difference here. There was not a statistically significant difference in the difference in reaction times between incongruent conditions for the Mozart or the incongruent and congruent conditions of the range time. So we do not have the interaction. So our hypothesis was not supported. And I think you can see it pretty clear here. You can also kind of see what I was talking about earlier, how in general, people were slower for the Mozart condition, for the range times condition. You can see these bars are taller than these bars. But that difference was not significant. The main effect of type of sound was not significant. So people were not significantly slower for Mozart than they were for the range chimes condition. So do we have any questions or comments about how to perform the data analysis, how to interpret the data analysis, what these graphs are showing? All right. Okay, so now the second half paper, we're ready to write that now. We have all the participant info and all the data compiled. You know what statistical analysis to perform. You should know how to perform it. You should know with pretty good confidence how to interpret the output. And I've also told you what you're going to find, though I haven't given you the specific numbers. So we're basically ready to write the second half. And I've mentioned it will be due November 1st, Sunday at 11:59 p.m. This link should go to the instructions. I'm not going to be reading them right here, but let's just make sure that this works. Yep. So here is the Group Project 1 second half. I finished it this morning. I think it's pretty good. I haven't had a chance to go through it with a fine-tooth comb and make sure that there are no typos. So if you do see anything weird or typos, please bring it to my attention. I always appreciate that, and remember the students that are helpful in that regard. Though I think it's pretty good. It should be pretty good. I was pretty careful, though I didn't have a chance to do fine editing of it. So it's there. It's also posted on Group Project 1 folder on Blackboard, the exact same information. So the things that you're going to have to do, you'll be writing out of these things. So I mentioned last time that I would show you a little bit more about APA formatting. This should go to the link. This takes you to an example APA paper. And I mentioned last time that there was more APA formatting errors than I was hoping to see in the first half assignment. And I will, for the second half assignment, I will still be grading for your formatting. It's not a huge part of the grade, but it's definitely something that matters. And you want the formatting to be right, because it will, if your formatting is wrong, it'll definitely hurt your grade some. And so use this right here to help you with formatting. Also, the lecture from earlier in the semester where I go into more detail about different aspects of formatting, including citations and headings. Those were the two big things that I saw errors on, citations and headings. So out of all of this stuff here are all of the parts of APA style paper. You guys have for first half, you already did the title page, and you already did the introduction, and you've already begun or done most of the references section. You've also typed the word abstract on the top of page 2. So the things that you'll be doing for the second half paper is you will be finishing the abstract, you will be writing the methods section, writing the results section, and writing the discussion section. There's kind of like 4 things. The abstract is pretty short though, because it's just 1 paragraph. So, basically 4 things that you're gonna have to do and write about them. And all of the information about what I want you to include is in these instructions right there. They're pretty detailed. I'll show them to you again. Pretty detailed. I basically go through each section, the abstract, the method, the results, and the discussion, and basically give you guidance, just like I did for the first half paper, of the types of things that I want you to be including. Pay special attention to the method section, because I'm requiring you in the method section to have 2 subsections. I've mentioned this before. Those subsections are the participants subsection and the procedure subsection. And I've given you guidance as to what I want you to include in those 2 subsections. Let's move on. I think I have, yeah. So I've actually shown you some of these exact slides before. You've seen this one before, but this is just giving you a list of the things to include in the abstract. I give you a similar list in the instructions for the second half paper for group project 1. So here's a typical method section. As I said, you're only going to have to do the participants and the procedure. Please note right here, I say that the apparatus or material section, this is not needed for your paper. You will need to do the participants and the procedure section. Once again, I've given you pretty specific information in pretty specific guidance as to the type of things I want you to include in each of these subsections in the second half instructions. So the results, I want all of these things. Our test statistic is F. When you're reporting F, this has actually been a mistake that I've seen in some recent labs that people are still making. When you are reporting an F value, you will be reporting 2 different degrees of freedom, usually a small number and usually a bigger number next to it. So don't forget, you'll be reporting 2 different degrees of freedoms. If you have questions about the degrees of freedom, don't hesitate to set up a meeting or email me. I can help you with that. And then the p-values for each statistical test. You do not have to report effect size. I mentioned that before. That would be eta squared. I did not check that box in SPSS, so you do not have to report effect size for group project 1 second half. So yeah, it says here when you're describing the group differences, you'll want to report the means and the measure of variability. That's what the figure does. The figure that I showed you earlier had the 4 condition means. That was the height of each bars. It also had little itty bitty error bars that I made, and I used Excel to put those error bars onto the graph. I will show you how to do that Monday. And basically exactly what I show you to do for that graph is what I expect you to include for your figure in group project 1 second half. Those error bars are the measure of variability. And those error bars were called within subject standard error. You've probably not learned how to make those yet, so I will show you how to make them Monday. And then you will want to explicitly mention the figure somewhere in your results section. Usually our authors will say something like, the main effect of congruency was significant. The main effect of type of sound was not significant. The interaction was not significant. Then the next sentence would be see figure 1. Something like, and I put a similar thing. I said see figure 1 right here. And that's where the means and the within subject standard error are shown for each of the 4 conditions in our study. And then finally, the discussion section. I give you, for all of these sections, I give you real specific information about the type of information to include. There's really 2 main paragraphs that I expect to see in the discussion. Basically, the first one will be short, which is state the basic findings that we had, and then discuss whether they supported our hypothesis or not. Ours provide basically only partial support for our hypothesis. They support our hypothesis in the sense that we expected Stroop interference. We found that. Participants were slower in the incongruent condition than the congruent condition. But they did not support our hypothesis in the sense that we expected the interaction. We expected Stroop interference to be bigger in the range times condition than we did the Mozart condition. So you'll want to say that in the first paragraph. And then the next paragraph will be basically saying the limitations, qualifications of our study, and then talking about future directions. So our study pretty obviously has some really obvious limitations. And I've listed some examples in the instructions for the second half. So you can kind of use the ones that I suggested. But I also mentioned when you are doing the discussion, part of your grade on that will be, are you coming up with good explanations for why we didn't find what we expected to find? And so I'm looking for you to be creative there and insightful, reflecting on what we all did together on group project 1 and why that might have made it so that we didn't get the hypothesis that we expected. Or you might reflect and realize you might disagree and be like, Well, I see these data and I just believe that listening to Mozart does not improve people's executive function. And then you can put that as well. That if you're going to put something like that, which I think would be cool, you might want to include a citation that kind of supports that claim. Any questions or comments about the different sections here? Oh, one thing more I want to mention I didn't say. My suggestion is that for the second half, you add these sections, the abstract, the methods, the results, and the discussion, add it to the first half papers that you already did, so that when you're done, you're going to have one entire correctly formatted APA manuscript. So I only will be grading, I'll be looking at the formatting of everything. So I will be looking again to make sure that the formatting of your headings is correct, but I will not be reading the introduction again. I will not be reading the introduction or grading. For instance, I will not grade the introduction. No points will be allotted for the second half to how well you state with clarity the hypothesis in the introduction. I'm done grading that stuff. But I will be looking at the overall formatting. So things like, are citations correct? Are all of the citations that appear in the text, do they also appear in the reference section? Are the headings correctly formatted? So that is it. I think that's all I have to say. And no one seems to be wanting to ask questions, so I'll just continue on. And so yeah, the next thing to talk about now is the EEG stuff that we started talking about last time. So here, I showed you this thing last time, so we're now segueing back to the physiological aspect of this course. The second half of this course, we'll be talking a lot about physiological measures where sensors are placed directly on a person's body, and you're measuring some type of biological activity with these sensors. In EEG, electroencephalogram, we're measuring electrical activity on the scalp. Presumably, most of the electrical activity is coming from the brain, though I should mention some of the electrical activities actually is coming from muscles in the face and around like your ears and stuff. There are muscles, and the muscles emanate electrical activity. So some of the electrical activity which is being picked up on the scalp probably is coming from muscles, not the brain. But it's generally assumed that most of the electrical activity is coming from the brain. And we talked last time about how in EEG studies, it's common to take the raw EEG signal and break it down into different sinusoidal components, which I'm going to go into more depth today as to exactly what that means. Those different sinusoidal components, to review, gamma is considered a high frequency component. Beta is high, but it's relatively lower frequency than gamma. Then alpha, then theta, and then delta is considered the lowest frequency. So to go into more depth, I talked about this last time. Fourier analysis is basically how you do this, how you take a raw EEG signal that's measured from some electrode on the scalp, and the raw EEG signal just kind of looks like random electrical fluctuations. And then you basically can apply mathematical formulas to that raw signal to decompose it into these different frequencies, into these sinusoidal waveforms of different frequencies. And that's what Fourier analysis does. And so this is an oversimplified kind of way of thinking about it. But for our purposes, we don't have to be experts on the mathematics of this, because the tools that exist can do a lot of this stuff for you without you having to understand the mathematics at all. But I want you to understand conceptually what a Fourier analysis is doing. So this blue-green box right here, this represents the raw EEG signal. So from some electrode on the scalp, we're just looking at those fluctuations of the electrical activity. And that's what this is right here, just the raw electrical activity picked up by electrode. What the Fourier analysis does, though, is it decomposes this complex waveform into the different sinusoidal components. So for instance, this waveform right here can be thought of as the sum of this component plus this component plus this component. The low frequency component, you can kind of see the outline of it right here. And then there's like the high frequency component are these little things right here, like right in within each of these things, there's high frequency component. And so this plus this plus this, these are each of like the frequencies. So this would be maybe like, these are just made up, maybe this is delta, maybe this is alpha, and maybe this is gamma. If you add all those up, each of those sinusoidal components, when you add them together, equals the complex waveform, equals the electrical signal that is measured with electrodes on the scalp, the raw EEG signal right here, and these are the different sinusoidal components of that EEG signal. So I've added something. So I've added, you guys, unfortunately, we didn't get to do this lab, but earlier in the semester, I went into Fraser and with my assistant, we made a recording of myself being a participant, showing you basically this study, which is described right here. So we talked in particular about alpha. I've already shown you this whole slide before, though I did add the link right here. And I mentioned that the frequency that we're going to talk the most about in this class is the alpha band. So let's go back real quick to this. The alpha band is this middle frequency right here. It's called 8 to 12 hertz here. And I noticed that in the EEG Review 1 article that you have a quiz that's due tonight, they call the alpha band 8 through 14 hertz. Still in the same ballpark. So alpha band is basically 8 to 12 hertz, maybe 8 to 14 hertz, depending on who you're talking about. And that is the frequency. That is the wave, the sinusoidal wave component that we will talk about the most in this course. And one of the studies that's used in a laboratory that we would do if we weren't, having to social distance right now is what is described right here. A study where there's only one factor, very easy, and that factor only has 2 levels. And the factor would be position of your eyes. And your eyes can either be closed, and we're measuring EEG, and then we're looking at the alpha amplitude when your eyes are closed. And then you open your eyes and you measure alpha amplitude when your eyes are open. And the question is the alpha amplitude different depending on whether your eyes are open or whether your eyes are closed? And the answer is yes. It's one of the reasons we focus on alpha so much in this course is because this effect in particular is very strong. It's very easy to find using the EEG. So I will show you it right here. You should be able to hear my sound, but I guess it really doesn't matter too much if you can't. So that's me.
Okay, so biopack is going.
Okay, so I want to point out things as they're occurring. So for one thing, there's me, and that's my assistant, Alex. And what you're seeing right here, I only have one electrode hooked up on my head. There's a lot going on there, but there's only actually one electrode hooked up to my head right here, and it's over my occipital lobe. So back here in the back of my scalp, because that's where these alpha effects tend to be strongest, is over the occipital lobe. And what you're seeing right here is the raw EEG waveform. So I mentioned, it kind of just looks like random fluctuations, and that's what you're going to see here. So what you're seeing here is just the raw EEG.
Eyes open. Eyes closed. Eyes open. Eyes closed. Eyes open. You're done.
Not bad.
Okay, so before I go to, it's going to go to a new screen here in a second. I just want to point out a couple things. Please note, so at the beginning of the experiment, my eyes were, closed and open, closed, open, closed. Yeah. So Alex, at the beginning of the experiment, my eyes were closed, and Alex said, open your eyes. And then she pushed a button on a keyboard that sends a marker. And it basically says right here, this is when the participant's eyes were open. And then when she said, close your eyes, she pressed the marker again. And that basically says, this is when the participant's eyes were closed. And then she said, eyes open again. This is when the participant's eyes were open. And close your eyes again. This is when the participant's eyes were closed right here. And then I guess she pressed a marker at the very end when she said, you're done. But that's how data analysis in EEG works. You have these markers which divide up the points and times depending on what condition you're in. These are the eyes open, eyes closed, eyes open, eyes closed, and then this is the end once we're already done. The next thing you're going to see is that in this particular program, BioPack, you just press we're done, and then BioPack does the Fourier analysis for you. So what you're going to see here in a second is 4 or 5 different sinusoidal components. And basically what BioPack does is it breaks down into different sinusoidal components, and you can see how does amplitude change in these different sinusoidal components between the two conditions, eyes open and eyes closed. So let's kind of watch here.
Eyes open, eyes closed, eyes open, eyes closed. It's always that one that's the biggest. You can kind of visualize it.
Yeah, that one looks bigger
Yeah.
visually.
So we'll probably end up keeping this one call today.
Okay, so I want to point out some more things. Right here, this is alpha. And this is where the eyes are closed, and you can see the alpha amplitude is really small. When the eyes are open, the alpha amplitude is big. When my eyes are closed, the alpha amplitude is small when my eyes are open. The alpha amplitude in this particular trial, my eyes are closed. The alpha amplitude is kind of big, and then the alpha amplitude is small right here. So you can actually see it. One of the reasons I like this demonstration is because the effects are so strong, you can actually see it when you're looking at the alpha amplitude. Here's beta, here's delta, here's theta. So I guess gamma, they don't do the Fourier analysis for you for gamma, the gamma frequency band, but they do alpha right here. And basically the analysis involves measuring the amplitude during this whole part. That's the eyes closed. And then the eyes, this is the eyes open, eyes closed, eyes open, eyes closed. And you basically compare the amplitude between those two conditions, eyes open and eyes closed. And we always show it's quite a strong effect that the alpha amplitude is bigger when your eyes are closed, which is what it says right here. Alpha amplitude increases when your eyes are closed. So this is kind of reviewing what we talked about last time. See how we're doing on time. Okay, so let's talk now. So because alpha has these properties, it's been suggested that alpha amplitude increased when you're relaxed. So you're awake when you see these alpha, but when you're relaxed, that's when you start seeing alpha amplitude increase. That is when your eyes are closed, that's when you see alpha amplitude increase. So it's been suggested that when you're really paying attention to something, when you're really focusing out on something out in the world, your eyes are open and you're really engaged with the thing, that's when you would expect to see alpha amplitude being the smallest. And so there's been various studies which have kind of looked at how does alpha amplitude, how is it correlated basically with different aspects of behavior. You would expect, for instance, that when alpha amplitude is really small, that your ability to respond to something out in the world would be faster, because that means alpha amplitude seems to mean you're highly engaged with what's ever going on in the world. If you're highly engaged with what's ever going on in the world, then your ability to respond to some type of stimulus in the world should be faster. So one prediction based on this kind of basic findings with alpha is that when alpha amplitude is small, you would expect people's reaction times to be small as well. Quick reaction times when alpha amplitude is small. And conversely, when alpha amplitude is big, that means you're kind of relaxed. That means you're not really trying or engaged with what's going on in the world. When alpha amplitude is big, you would expect reaction times to be big or slow as well. So here I'm going to present you some classic studies in this regard. They are presented in that green textbook I showed you previous lecture. It's a little bit outdated now, and you can see these studies are old. So they're classic studies, so they're pretty famous, I would say. But that's like 60 years ago. That one's like 50 years ago. So these are pretty old studies that we're talking about, but they're interesting effects nonetheless. So we'll go over these a little bit today. So a term which I need to tell you before you can kind of understand these studies is alpha event-related desynchronization. Event-related desynchronization. All that just means that alpha amplitude is decreasing. You could kind of, event-related desynchronization just means that alpha amplitude decreases. And that's a term which they use in these articles which I'm about to talk about. And another term, which you might see sometimes in like articles you might read, is called alpha blocking. Both of these terms, ERD, event-related desynchronization, and alpha blocking, just mean that alpha amplitude is small. So here's the 1959 study. So to talk about this, I need to talk about the 4-period paradigm. So the 4-period paradigm, people are given a warning signal, and that warning signal is usually just like a beep. And what that warning signal is warning you about is that there's going to be a target stimulus in the near future, and your job is to respond to that target stimulus as quickly as possible. And generally, if they're in studies, sometimes they'll give you a warning signal, and they find that giving a warning signal and then there's a target a little bit later, that allows the people to respond to that target a little bit more quickly. It allows them to prepare mentally and physically for that target which is about to occur, which allows them to have slightly quicker reaction times compared to studies where there's no warning signal at all, in which case people respond to the target relatively more slow. So this question I've kind of already mentioned, do the faster reaction times, do they correlate with alpha amplitude or alpha event-related desynchronization? So to talk about the specific study, Lansing, 1959, I need to talk about variable 4 periods, because that's what they use here. On any given trial, you're given a warning signal, but the amount of time in variable 4-period studies between the warning signal and the target is variable. So the thing which is variable in variable 4 periods is the amount of time between the warning signal and the target. And the general finding that they observed, that is talked about in this Lansing 1959 study, is the more time that passes after the warning signal, generally the faster the reaction time tends to be. So in some trials, there'll be the warning signal, and then 200 milliseconds later, there'll be the target that you have to respond to. On some trials, you'll have the warning signal, and then 500 milliseconds later, there'll be the target that you have to respond to. The general finding is that people tend to respond more quickly when the target occurs 500 milliseconds after the warning signal than when it occurs 200 milliseconds. And the explanation for this is that once that 200 milliseconds passes by, you know with high confidence that the more time that passes, the more confident you are that the target has to occur very soon. The warning signal says in the near future, you know that this signal is going to occur. You know that the target's going to occur soon. The more time that passes after that warning signal, the more confident that you are that it has to occur soon. It has to occur sometime soon. And the more time that passes, the more confident you are that it's going to be occurring very soon, the quicker your reaction times tend to be. The question, once again, though, is do faster reaction times for longer 4-period durations, do those people that are fast, do they show smaller alpha amplitude? Are they more engaged with what's going on in the world that allows them to respond to the things in the world more quickly? So this is super old school looking figure, but it kind of helps to illustrate the things I was just saying verbally. This is kind of a schematic diagram of a study like I just described. So right here this is the warning signal. S, this is like the stimulus. So this is what I was referring to as the target. So this would be the long 4-period duration. This is like 500 milliseconds right here. And the R is the person's response. So this distance right here is their reaction time. So warning signal, long 4-period duration around 500 milliseconds, the target stimulus, and then right here is the participant's response. Down below, this is this is the warning signal, and this is a short 4-period duration, only around 200 milliseconds. And then there's the target right there, and then the participant's response right there. One thing I want to point out is kind of hard to see, but you can see that the response right here, there's a bigger reaction time right here. The distance between the target and the response is relatively longer right here because the 4-period duration is smaller. Over here, where the 4-period duration is relatively longer, if you look at the reaction times, it's a little bit shorter. So over here, shorter reaction time right here with the long 4-period, and right here, there's a relatively longer reaction time for the short 4-period. They were measuring alpha amplitude. That's what these waves are right here. They were measuring apparently with 2 different electrodes, occipital and over motor cortex, so that presumably was over the frontal lobe or the frontal parietal area up here. We're most interested in the occipital phenomenon, which I presented, right here. So they showed basically that they were looking at, they basically divided up people's trials into whether the participant was showing alpha blocking or not. And so you would assume alpha blocking, once again, means alpha amplitude is small. Alpha amplitude is blocked. That means the alpha amplitude has been decreased. It is small. And you would make the prediction that people who show alpha blocking will show faster reaction times. People whose alpha amplitude is small should respond to the target stimulus more quickly than people whose alpha amplitude is big. So it's a little bit of a complicated study, and actually the way they present the data is really complicated by today's standards. So I apologize for that, but this is 60 years old, and I always think this is like when Marty McFly goes back into the past and Back to the Future. That was 1955. So this is like the same point in time this is old stuff. And they definitely didn't have Microsoft Excel to make graphs. I'm sure making this table was quite a lot of work back then, whereas today it would be very easy. And the data are not presented in a way which is particularly easy to understand. So I'm going to talk about it a little bit today. Then I'm going to move to something else, and then we'll actually come back and talk about this after you've had time to consolidate this information. We'll come back and talk to it a little bit Monday. But I want to point out that here they say alpha not blocked. So this means basically these are the people whose alpha, when they were measuring the alpha activity, the alpha amplitude was big. And if you look at their reaction time was on average 255 milliseconds. And then these people right here, their alpha was blocked, meaning their alpha to alpha amplitude was small, and their reaction times were also small. And so this study does suggest that when alpha amplitude is small, that it basically causes you to respond to the environment more quickly. So I want to come back to this. There's a lot going on in the study. Alpha amplitude is kind of a tough concept. So I'm going to end this talking about this article right here. We'll finish talking about it next time, and then we'll talk about another article next time, Levitt 1968, which kind of contradicts Lansing. So there's these 2 studies, they're both old, and they actually kind of provide it's a controversy. They kind of did 2 different studies, and one study suggests, alpha amplitude correlates with reaction times. The other study says, nah, it doesn't appear that alpha amplitude actually does correlate with reaction time. So this is a controversy, and we'll finish kind of talking about this controversy next time. What I want to do before we get out of here today is talk about EEG Review 1 briefly. So you have a quiz tonight, and I'm feeling in a nice mood, so I'm going to kind of address some of the questions which are on the quiz for tonight. Not all of them, but some of them. And these are the figures which I happen to ask questions about for EEG Review 1 quiz, which is due tonight. So there's kind of 2, this is a review article, means it describes and summarizes the results of a bunch of different studies. And right here, this is the short-term memory study. There's a question about the short-term memory study, and that's what this is referring to right here. And so in this study, it's short-term memory, so you guys should know a little bit about how that works. You're given a list of things to remember, and that list either had 2 things. So imagine like 2 letters you have to remember, or 4 letters, or 6 letters. Maybe it's numbers, but you have to remember either 2, 4, or 6 things. And they say, remember these things right here. And that's what the memory set is right here. So this is like the beginning of the trial. And you notice how the y-axis here is frequency. And so, and you notice the frequencies they're kind of zooming in on here is the alpha band. Though they include some stuff a little bit below the alpha band here. So alpha band usually is like 8 through 12 or 8 through 14 hertz. And so the entirety of the alpha band is shown on the y-axis of this figure. right at time point 0, this is when the memory set, the items that you have to remember, are being presented to the participant. And then there's a delay time of 3 seconds. And this is presumably this is the time during which, if you're using your short-term memory, you're kind of rehearsing that information in your head over and over and over again so you don't forget. And what you see here, this red splotch right here, means that there is a lot of high amplitude activity that's occurring at around 11 hertz. So 10 hertz would be right there. 11 hertz seems to be about right there. So at around 10 or 11 hertz, there's a whole lot of activity that is within the alpha band. There's a whole lot of activity that occurs when you are rehearsing that information in your short-term memory. And then finally, a probe will come. A probe is a single letter or number, and your job once the probe comes is to indicate, is this probe right here, was this in the original memory set that we showed you 3 seconds ago? And you notice that once the probe is shown, they're no longer having to rehearse that information in their head, and the alpha amplitude goes away. So here's a situation that basically when you are rehearsing something in your head, trying to remember it, there is an increase in alpha amplitude. An increase in alpha amplitude. The really interesting finding, which pertains in particular to the question for tonight, is what happens to alpha amplitude the more items that you have to remember? And that's shown here. I keep saying amplitude, but the y-axis here is actually power, but power is just amplitude squared. So amplitude and power are highly related to each other. Power, once again, is just whatever the amplitude is squared. That's what power is. So we're looking at amplitude squared, or power, here on the y-axis. And then right here at around 10 hertz or 11 hertz or so within the alpha band, you can see the different conditions: 2 items, 4 items, 6 items, and you can see quite clearly that the more items that you have to remember, the higher the alpha band activity. So once again, this is quite consistent with what we were just talking about in the previous studies. But we'll kind of reflect on this stuff more next week. This makes sense. It makes sense that the more items that you're kind of rehearsing in your memory, the bigger the alpha amplitude tends to be. We'll talk more about why this makes sense theoretically next week. And then here is the other study from EEG Review 1. So this is a study where people were paying attention. They were cued to pay attention to either the left side of the world, the left hemifield, or the right side of the world. And the researchers were measuring alpha amplitude. You can see it, they actually call it alpha amplitude here. They were measuring alpha amplitude in the right hemisphere occipital electrodes, and they are also measuring alpha amplitude in the left hemisphere occipital electrodes. And they were looking at how amplitude changes in each hemisphere, depending on which hemifield you're paying attention to. So remember, kind of a theme, or one of the things we've talked about many times in this class, is the tendency for information in the right side of the world tends to be processed more heavily in the left hemisphere of the brain. Information from the left side of the world, the left hemifield, the left visual field, tends to be processed in the right hemisphere of the brain. And there appears to be kind of a neural correlate of that in terms of alpha amplitude. And so I'll just show you one of them right here. So here we are looking at the right hemisphere electrode. The green trace is when the participant is paying attention to the left side of the world. The red trace is when the participant is paying attention to the right side of the world. So remember, the right hemisphere of the brain should be particularly interested in the stuff in the left side of the world. And that's basically what we see right here. You can see that in the right hemisphere, when you are paying attention to the left visual field, alpha amplitude is relatively smaller than when you're paying attention to the right hemifield. So in other words, kind of the general principle, when you're measuring alpha amplitude from the occipital lobe, alpha amplitude will tend to be smaller when you are paying attention to the contralateral side of the world, part of the world which is opposite from the hemisphere you're recording from. Alpha amplitude tends to be smaller when you are paying attention to the contralateral side of the world. You see that right here. The right hemisphere, alpha amplitude is smaller when you're paying attention to the left hemifield, and the converse is true down here. when you're measuring from the left hemisphere, alpha amplitude tends to be smaller when you're paying attention to the right side of the world. So alpha amplitude is always smaller in that hemisphere when you're paying attention to the opposite side or the contralateral side of the world. So this is pretty dense stuff today, so I'll definitely call it a day there. We'll call it conclude things here. We'll kind of review some of the stuff. We'll finish talking about those 2 older studies next time. Then we will move on talking about event-related potentials, and I'll show you how to make figures Monday for your group project 1. So I do believe that I have a group that wanted to meet with me after class, and we'll do that once I stop recording. And I want to remind all of you guys, if you want to set up appointments to meet, to talk about your study idea for group project 2, I think it will be informative, and it'd also be pretty fun. I think kind of brainstorming ideas is one of the funner things to do in science. I have a chat right here. Let me see if I should answer this here. This one student is saying that the EEG Review 1 article was not loading properly. I'll have to take a look. I'm going to try and download it myself.
I had that same problem.
Okay, so multiple.
And it's like you have to enter like, I don't know, something, but it doesn't work. I just Googled it, and the PDF popped up online. So I just copy and pasted the article name.
Huh, interesting. Okay, yeah, I'm seeing there's like 3 people now agreeing with this. So
I also
It worked for me. I was able to take the quiz and I saw the article. It was fine on, at least for mine.
weird. So there's
it has to do with the browser. It won't work on Chrome, but if you open it up on Edge, it works.
weird. God, that's
Yeah.
really weird.
I've had that problem with a lot of articles on Blackboard.
I used Chrome. I don't know what's up with it then.
Okay, so but wait, which browser works? Does Firefox probably works because that's what I used almost all the time is Firefox. So Firefox would be something to try. But yes, if you're having troubles, I'm sorry to say I didn't know that Blackboard was having problems with that. If you're having troubles downloading the article, one potential solution is to try different browsers. If you still have trouble, you can always email me, and I'll email a PDF copy. I'm sorry that that's happening. I will look into that today. I'll also point out EEG1 and EEG2 quizzes. I will be posting those later today as well. Yeah, thanks for bringing that to my attention. I'll stop recording now.